Where's everyone else? Lucy. Hi, Sierra. Hi, Katie. If anyone has questions as everyone's getting on, let me know. Hi, Fatima. Okay, I'll get it for you. Just give me another minute or so. Where's your water bottle, please? Here, just use mine. Here. No, here. There's no dishwasher. This is right for the one. Go ahead. Go finish your stuff. What? Are you done? Did you ready? You're trying to figure it out. Just a minute until I check it. Okay. Hi, guys.
Hello.
How's everyone doing?
Good morning.
Morning.
Good morning, Dr. Kaminsky.
Morning.
Hi, Dr. Kaminsky.
Hey, who was that?
Blossom.
Who was that?
Blossom.
Oh, hey, Blossom. How's everyone doing? Are all still hanging in there? So as you guys, since there's a decent amount of you guys on there, I put another topic up on that question and answer forum. So let me share my screen and just so that you guys you could do this now as we're as I give it another minute or two. So course question and answer forum, I put up attendance for today just to post a question or an answer. Let me get this out of here. Post a question or an answer. Oh, oops. Oh yeah, post a question or an answer to a question pertaining to the cell signaling chapter. And it can't be one of the homework questions. It has to be something else. All right, you don't need to do that right now, but it's there. Just want to make everyone aware. Who else? Just see who else came on.
Good morning.
Morning.
Hello.
Hi. So I was just telling everyone for attendance for today, you need to post a question or an answer to a question for attendance for today, 422, okay, pertaining to cell signaling. You don't have to do it right now, just, by the end of the day today or whatever, okay? Anyone have questions about anything before we get started? No, nobody right now?
I do.
Good.
Never mind, you just showed it. I was going to ask when is the final exam review due, but I just saw it said the 29th.
Yeah, I just changed it because I had it due the 27th. It's due next Wednesday, the 29th. Okay?
Okay, thank you.
Yep. What's up? Okay, then do something else now. You can start something else. No. All right, sorry. Okay. All right, so just some reminders. So that was one of them. So the final exam review, the completed final exam review sheet is due next Wednesday, okay? Obviously then you need to review all the material at least, once, so that we then can have a good review that day for the final. If you don't review the material, it's hard to ask questions about it, right? So make sure that that's done by then. And then you guys come, to Wednesday's Zoom meeting with questions or specific things that you want me to review a little bit more, okay? And also helpful to post questions ahead of time in the question and answer forum. I mean, not just right ahead of time for next week, but really anything that's going to come up about the final exam, anything you have questions about, post them there, okay? So that I can see them ahead of time. Anyone have any other questions kind of about anything that's due or anything else like that? Just general questions? The one other thing I think reminded you about this. This mTOR paper summary is due on Monday. I will post a recording to introduce that paper for you guys, okay? I was gonna do it now, but I feel like then it's too many things at once. So I'll post a recording, like I said, just to introduce that, and that's due Monday. That's obviously the last paper summary that you have. I'll also, in the next day or two, try to get caught up with grading. I have to grade your lab reports, obviously. Those are the big things. And then the other thing is this PCR assignment. So I have a bunch of things to grade. So I'm going to try to get caught up on those assignment gradings, so that hopefully coming into next week or by next week, I'll have everything graded. The only thing that will be left would be the final exam. All right. So that's my goal. Hopefully that will happen. So for the PCR, so first thing, PCR lab assignment, was due already, right? And so I just put up the answers to the PCR assignment. I gave everyone kind of a couple extra days to submit if they hadn't, because I told you I was going to post those answers. So I will grade that assignment, and it's going to be based on, these answers here. So I wanted to make sure that you guys had the answers. So what I did is, it's the whole PDF. But if you go to where the questions are, you can see my answers inside that PDF, okay? And so, like I said, I'm going to be grading this for accuracy, right? It's not like the homework questions where I kind of just grade them as long as we made a decent attempt at them and it's not just completely crazy. I pretty much give you full credit. But for these, I really am going to grade it in terms of accuracy, because really all of this information is in the text, okay? I guess a couple things, and I did introduce this and talked a little bit about the PCR process itself, and it does ask you a little bit about that, I think, in the second set of review questions. But I guess what if somebody, right? I mean, I have it here, right? What does PCR? What does PCR stand for? Who knows? Who remembers what PCR stands for?
Polymerase chain reaction.
Yeah, polymerase chain reaction. Thank you. And what's the purpose of doing PCR, right? I mean, and you can think about it in the context of this was kind of with DNA, like forensics and stuff like that. And so why do scientists need to do PCR, right? What's the function of PCR? What does it do? I tell you that there.
Usually there isn't enough DNA, so it allows you to replicate it.
Yeah.
In order to create more samples to be able to test it out.
Okay, good. Yeah. I mean, so again, you can extract DNA from what? I mean, what are some things that you can get DNA from? So at a crime scene, where do we get DNA from?
Saliva.
Yeah,
bone,
saliva, hair,
blood.
right? Really anything. Blood, right? But that amount of DNA that you can extract, especially if it's just a tiny amount or in a little piece of hair or whatever, you're not going to get much DNA, right? Not enough DNA to study it or to analyze it. Okay, so you have to use that PCR to amplify it, right? And make more of it. And so, but the one thing about the PCR, does it make more of all the DNA that's there, or what does it do? And this is sort of alluding to it right here. Does it amplify that entire, so you have, think about it, it's all the DNA, I mean, if you think about any cell in your body, all of your genomic DNA is within that nucleus, right? So does PCR amplify every single part of that DNA or no?
No.
Yeah.
No. So what does it do?
A specific strand.
Yeah, a specific region of the DNA, right? And how does it do that? How is it able to only amplify a specific region? Or target sequence, if you see here, right?
Yeah.
What are these?
The primers.
Yeah.
So
The primer.
Yeah. So
Like, does it just cut that specific strand that it wants and then replicates it over and over again?
it doesn't cut it,
Like, not
it
cut, like,
targets
not
it,
like
right?
Yeah.
So these primers, right, just like we talked about primers and DNA replication in general, right? Remember, DNA replication always has to start with a primer, right?
Mhm.
So what you do when you do this in a tube is you're adding primers that are specific to your target sequence, right? So if you want to just target a specific gene, here they're targeting, you know, this specific area of the DNA that they know will vary between individuals, right? Those primers every time will recognize that specific target sequence, right? And then that's what will be amplified. So the rest of the DNA doesn't get amplified, only that target sequence between those primers, right? All right, and then once you make enough of it, right, then you can actually study it. You can run it on a gel. So this ties into our electrophoresis, where we talked about the electrophoresis, right? Now you can take your products from the PCR and you can run it on a gel and see how they differ, right? And so in this case, when we're running things on a gel, right, we're sort of looking at the different bands that are formed, right? And so you can then distinguish between individuals based on these bands that you obtain, right? And so what are these bands here? What do they correspond to?
Base pairs? Like,
Well, yeah, I mean, there's base pairs. Imagine you're basically loading up here. Imagine this is a gel, right? You're loading your DNA here, and then you're putting it right in the buffer, attaching it to, you know, electrical current, and then you're letting that DNA run down, right? And remember, for the PCR, you're just putting these PCR products on there, right? So you've amplified these specific regions to look at these differences in alleles, right? And so you're looking, so it's basically sort of the genotype of these individuals at this specific locus in the DNA, right? And so here, right, these are these Th1 alleles. Okay, so there's all these different ones, and basically each person will have a different combination of these alleles, right? So this is showing you for suspect A, it has this number 5 allele and the number 3 allele, right? Suspect B has the number 6 and the number 10, okay? Now, in forensics, you're not only gonna look at this locus, right? They would do multiple loci like this that would help determine a sort of DNA fingerprint, and to determine what individual matched their, you know, whatever, their sample that they had or what have you, whatever they were investigating, okay? So does that make sense? So you need to run these on the PCR products on a gel in order to visualize what you have, right? So just running the PCR is going to amplify those regions that you have, but then you have to then be able to sort of to analyze that, okay? And so in this case, we're using the gel to analyze that, to visualize what products did we get, and then we can compare between different individuals. Does anyone have questions about that?
No.
Any other questions? That's about the components, right? And if you think about the components that are in here, and we think about DNA replication, it makes sense, right? One thing is, what about the polymerase that's in PCR? What type of polymerase is it?
It's from like TAQ top.
Yeah, TAC. So it's called TAC polymerase because it's extracted from special bacteria that can live in these really
Live in hot
high,
spring.
yeah, hot springs, right? These high temperatures. And so that TAC polymerase can withstand that 95 degrees that you need to heat the DNA to in order to denature it and separate the strands, right? So if you use a normal polymerase, like say a polymerase from a human or whatever, just for comparison's sake, and then you heated it and you put it in a tube and then you heated it to 95 degrees, it would destroy that polymerase and it wouldn't be able to work, right? So the process of PCR is really dependent on this TAC polymerase, right? And so if we didn't have this heat stable polymerase, this wouldn't be able to happen, most likely, right? So that's one of the key components to it. All right, does anyone have any other questions? Like I said, I'll be grading these. I posted these answers so that you guys can see what answers I expected. And then we'll go from there. If you have a question about your grade or something like that, just let me know. But I just wanted you guys to see that those answers are there, okay? Anyone have other questions about it? No, we're all right with that? Okay. All right, so let me go back. Okay, so like I said, those answers are posted here. And that's pretty much most of it. Like I said, I'll catch up on grading. The other thing is, let me just pull up the review sheet and just look at those cell signaling, some of those questions there. I'm hoping that most of us had a chance to listen to the cell signaling recording. If you haven't, please do. Make sure you're completing and filling out your review sheet as you go through and then answering those review questions. So if we want to just kind of go over some of these review questions just to kind of start hitting some high points in this chapter, let's do that. But again, you guys need to ask questions or let me know if there's something that needs more clarification. The concepts in this chapter are difficult, right? This is not an easy chapter. Like I say in the recording, you know, you could spend a whole semester or a whole year or even your whole career studying one of these specific pathways or even just one molecule in one of these pathways, okay? And a lot of them are targets for, you know, or therapeutic targets. For instance, for cancer, right? A lot of these signaling pathways are involved in cancer, and I say that a couple times in the lecture, okay? So again, when you're sort of listening to this and trying to study this, try not to get, I guess try to think about big picture as to why it's important to learn about these pathways, and not kind of get this in your mind, like, why the heck do we need to know about every, you know, all these different pathways? And again, remember, this is just scratching the surface in terms of the different pathways, signaling pathways that exist. All right. Any questions sort of off the bat about this chapter or I don't know, is there something that you, and again, you guys are going to put this in for attendance for today. We're posting a question or answering a question related to this chapter. And also, please, while you're going through these things and listening, any question that you have or anything that needs clarification, put it up in the discussion forum, okay? So then that way I see them and I can address them when we meet again next week, right? So again, reminder, if some people came on a little bit late, for today's attendance, you're writing a question or an answer to a question specific to cell signaling. Also, remember I have this other topic, final exam content questions. Anything that comes up, put it there, right? So everyone can see. So maybe if it's quick, I'll just answer it there, or I'll just, you know, bring it up at the next class meeting. If it's there and for some reason I don't bring it up or answer it, please draw my attention to it at our next Zoom meeting, okay? But let's try to keep things sort of organized here, all right? Any questions, guys? Any other questions for now? All right, so let's just look at the review questions. We'll sort of take it from there. And I know probably a lot of you, even if you did listen to this recording once, there's probably going to need to be sort of more studying that needs to go on for this chapter. So the first question is pretty easy, right? Describe the 3 modes of signaling of secreted molecules. So it doesn't just say list, it says describe. So what are the 3 modes of signaling for secreted molecules? What are these? These are the 3 modes of signaling, yes? For secreted molecules. So when we say modes of signaling, we say direct or secreted. What do we mean by direct?
Cell to cell.
Yeah, cell to cell. So, and a lot of times, so this is giving you an example of integrins and coherins, right? But basically, and sometimes some of these things are sort of involved in growth factors. Some of them too can sort of be direct, but a lot of this kind of direct cell to cell signaling has to do with too during development. So like during development of the nervous system, for instance, when pathways are being set up, if you think about kind of these networks that exist within the brain, a lot of that and how neurons find their targets uses this direct cell to cell signaling. So anyway, just to kind of give you an example, but I'm obviously not going to get into that, but what it is right, you can kind of think of this as like, you know, it's not a hormone or anything like that, or this is something that would be secreted from the cell, but in this case, it just sits on the surface, right? And this, then the receptor. So like I said, this happens a lot during development, not just during development of the nervous system, but when cells need to sort of find each other and find targets, they'll use this type of signaling, okay? but we mostly focus on the secreted signals here. Okay, so there's one example of a secreted signal, endocrine, and it's listed here. So what's endocrine? What do we mean by endocrine? Endocrine signals, how are they released? Or where do they go? They're secreted, but then how do they then reach their target cell?
The circulatory system?
Yeah, the circulatory system, good. Meaning they get secreted out of the cell, they enter into the bloodstream, right? Then they can travel long distances to reach their targets, okay? So that's, when you think of endocrine, you're thinking of glands, you're thinking of hormones, right? They're released into the bloodstream. And also think about long distance sort of communication, right? They can reach targets that are far away. All right, good. What about paracrine? What's our example of paracrine signaling? Paracrine. So this is endocrine, right? Released into the bloodstream. What about paracrine?
Produce different technologies.
Right? What about paracrine here?
Neighboring?
Yeah, neighboring,
So-
right? Is it released into the bloodstream? No. When we think about neurotransmitters, right, they get released into that synaptic cleft and then they just diffuse across that space and bind to a neighboring cell. They don't have long distance targets. Their targets are close. So good. Okay, so that's paracrine. So I hope you can see the difference between endocrine and paracrine. Then there was one other type of secreted signaling called autocrine that I mentioned. Okay, and so what about autocrine? What's that? What's happening there?
Checking outside.
Well, so this is a cell, right? This is a pretty, they could probably have some better diagrams. I probably could draw this cell. The signals released, it does the trick though, right? The signals released from that cell secreted, and then where does it bind? To another cell? Where's it binding?
The same cell.
Same cell, thank you. Right, it's binding the same cell, hence the term autocrine. And we gave some examples about growth factors, right? So the example that T lymphocytes will basically produce a growth factor that then will bind to themselves to promote their own proliferation, right? And so when you think about an immune reaction, right? Or if you think about injury, there are immune cells that basically the body wants to produce more of to help basically with whatever's going on, say at that injury site, right? Maybe to clear debris away, clear old cells away, maybe to secrete other factors that are going to attract other cells, but you can see how this might be beneficial. But it also contributes to cancer, right? Cancer cells find a way or know how to secrete growth factors that then stimulate their own proliferation, right? So if cells that you don't want to proliferate can stimulate themselves to proliferate, that's an issue, okay? So if you kind of think about an application of autocrine, right? So hopefully we all know the difference between those three, and we really could describe the differences, not just list, right? But explain, maybe give an example, right? I basically kind of give you an example for every single one of them. Any questions about that? No? All right. The next question says, asked about the basic mechanism of G protein coupled receptor signaling. And I think I kind of pulled this up quickly, or I showed you guys an animation of this. And so there is an animation. I think there might be two related to this. I don't know, but check Moodle. There's a couple animations, but, and in the recording, I really told you, like, we need to understand the G protein coupled receptor signaling, right? Obviously, again, I only scratched the surface with giving you some examples, but that was the one that we spend the most time on. And then I did give you other examples of other types of signaling pathways as well, okay? So it asked you about the basic mechanism of G protein coupled receptor signaling and asked about the second messenger, okay? So if we go to one of those diagrams, and I think it might be the same diagram that I pulled up before, and I got, obviously there's other information in between this topic and this one, so make sure you know that. The way the lecture is organized is it talks about the different types of signaling molecules, right? So mentions hormones, neurotransmitters, then talks about peptide hormones or neuropeptides, and then growth factors, right? Things like that. Mentions cytokines, and then goes through and sort of gives you examples of some of the signaling pathways for those molecules. Okay, so just so kind of, if you kind of keep that in mind to try to keep your mind organized as you move through that lecture, but that's sort of the way that it's organized, right? So if you're talking about like a cytokine, so what's a cytokine right here? What types of cells release cytokines? So immune cells in general release cytokines, right? And so anyway, I mentioned cytokines, and then later on showed you like an example of a receptor that a cytokine binds to, and then what happens inside the cell, that signaling pathway. Okay, so that's the way it's set up. We sort of talk about some different types of signaling molecules, and then give examples of how they may initiate a response in their target cell, right? When we talk about a signaling pathway, we're talking about what's happening in the target cell. Once that molecule binds to its target, what changes are being initiated inside the cell, right? So that's big picture. Does anyone have questions about that? Do we understand sort of big picture what we're talking about? And then, you know, obviously you got a lot of nitty gritty details to which, again, we're only just scratching the surface on, but any questions on that? So I sort of got on a tangent a little bit, but I was moving over here to talk about G protein coupled receptors, right? And couple things. So, and then we're pretty much going to end with this, but couple things here. So the G protein coupled receptors, and I point this out in the recording, right? If you kind of look up top here, and you're talking about a G protein coupled receptors, to which there are many, 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 many different types of G protein coupled receptors. But if you kind of want to make a generalization, what happens up top here and the beginning steps is usually similar, okay? But then what's going to happen downstream is going to be a lot different, okay? Depending on what molecule you're talking about here binding to what receptor, right? So this is showing you epinephrine binding to its receptor. And I also make this point in the lecture as well. There are also many different types of receptor subtypes, even for one ligand. So epinephrine doesn't always bind to the same receptor, okay? So there's differences. When you talk about like the opiate, the endogenous opioids, right? Or opiates like endorphin, okay? It can bind to multiple receptors as well. Those receptors are also G protein coupled receptors. They're going to sit on very different target cells than epinephrine receptors, right? So it depends on where the receptor is in terms of what cell it's on, but then also what type of receptor. Okay, so there's so much variety and complexity within this. Like I said, that's why I keep saying we're only sort of scratching the surface. But if we sort of just understand basics in terms of, all right, I know that things like this are going to happen when a hormone or when a neurotransmitter or when a neuropeptide like endorphin binds to its receptor, it's going to initiate a whole series of events inside that target cell. Okay, so for G protein coupled receptors, guys, again, what's up here is pretty similar. Okay, this is the same example I had given you a couple times in other chapters in terms of epinephrine binding to its receptor on a muscle cell, right? And then basically causing an activation of a particular enzyme at the end to convert glycogen into glucose. Okay, so you can kind of see the end result here. Now, this again is going to vary depending on what type of cell you're talking about, and I'll give you some examples. So here's another variation, right? Here's your G protein coupled receptor. This could be anything, right? And if you notice up top, do you guys see that this looks pretty much the same here? Right? By the way, what's the second messenger here? What do we consider the second messenger? So I ask you about that. So let's go back here. Where did I say what a second messenger is? Oh, where is that? Here. I sort of mentioned second messenger all the way back here. but let's talk about it when we're talking about the G protein coupled receptor. So I said a second messenger is a molecule that relays a signal from a receptor to a target inside the cell. Okay. So what is the second messenger here?
The epinephrine or
No. So the second messenger is actually, let me go back to that one picture here. We'll go, let's go to this one because I wanted to go through this. The second messenger is cyclic AMP actually. Okay, because this is still sitting, this adenylyl cyclase is still sitting in the cell surface, right? So basically this cyclic AMP is that second messenger that then is going to relay this message to some internal molecule. Okay, so for the G protein coupled receptors, the second messenger is cyclic AMP. Now, that's not always true, but for the most part, and for our purposes, when we're talking about these G protein coupled receptor pathways, if it's stimulatory, usually the second messenger is cyclic AMP. Okay, but that second messenger is going to be different if we're talking about different types of receptors. All right, so that's just something that's an important sort of I guess an important term to know in terms of, again, it's linking this sort of receptor or what's sitting in the membrane to molecules inside the cell. Okay. All right. So we only had a couple more. Go ahead.
Dr. C.
Yeah.
Oh.
Yeah.
So for the G protein,
Mhm.
in the picture, it has 3 subunits, but then only one subunit gets activated. So is alpha the only part of the G protein, or the other 2 subunits also G protein?
These are all part of the G protein. So the G protein has 3 subunits, beta, gamma, and alpha. But right, the alpha is the only part that becomes activated and then goes and basically kind of initiates this cascade of events. Beta, gamma kind of just hanging out here. But there are other instances where beta and gamma might have an effect. But in this case, when you're talking about stimulatory kind of pathways, it's really the alpha subunit that's going to sort of initiate the process. Okay? But
Thank you.
Go ahead.
I was just saying thank you.
Okay. The G protein itself, though, is composed of those 3 subunits. Okay? So for instance, I'll go back. Oh, no, that's my next one, I guess. That must be the next picture. All right, so I just wanted to point out something here before I move on. So what do you guys notice about this picture here compared to the one with epinephrine, right? And remember that final result was basically an enzyme was being activated. What is the end result here of this pathway? If this pathway gets initiated, right, or activated, okay, what's the final result? What's the end goal, I guess, of this pathway? What's happening? And you can just say in general, you don't have to say specifically, what's happening down here? 1 second, Mason. What's the end result of this pathway?
You're not done yet.
Mm-mm. What's this down here, guys?
transcription.
Yeah, transcription. So what's actually becoming, what's becoming activated, right? Essentially a transcription factor is becoming activated, right? Then it's it's able to bind to its target. And so it's showing you specific targets. Again, remember, we're not memorizing any of these details. We don't need to memorize what Cre is or what Creb is. But this is a transcription factor. How do you know that? It's binding to DNA, right? And it's initiating transcription. So a lot of these pathways will activate certain transcription factors or maybe inhibit transcription factors and will affect transcription, right? We talked a whole bunch about regulation of transcription, right? But we didn't kind of talk about, well, how is that sort of initiated, right? And so certain molecules, hormones, neurotransmitters, when they bind, neuropeptides like I said, endorphins or whatever, will actually affect transcription of particular genes. Okay. So again, I know this is complex and that's why we're not memorizing anything. we're understanding. So if we look at these diagrams, especially the ones that I point out on the review sheet, we should be able to understand what's going on based on the diagrams, okay? Like I said, we only have a few more minutes left. One other thing I just want to point out, oh, I thought, anyway, where's the, I want to see something, sorry. This, oh, maybe I don't have a picture of it. Yeah, don't worry about the whole, I was going to point something out about, so sometimes, okay, the, so you see this here? Because who was asking about the beta gamma subunits, right? If that G protein is inhibitory, I don't show you a picture of this because maybe I don't really want you guys to worry about it, but if you're curious, that G protein, you see this here, it says G sub I, can be inhibitory, meaning it's not going to activate things, okay? It's gonna deactivate or inhibit the target cell, okay? So in this case, once those subunits separate, right, the alpha subunit will inhibit adenylyl cyclase, and this beta gamma subunit will actually go and do something too. So sometimes the beta gamma does have a job, right? When we're focusing on those ones that activate, that's why we're focusing on the alpha subunit, because that's the one that's activating, okay? But so here's an example here where the beta gamma subunits will actually open potassium channels, which will help, which would inhibit the cells, okay? But again, if that confuses you, then just forget I said that, okay? Because, you know, it's hard to leave out so many details, and it's hard, it's hard not to put in too many. So, so anyway, that kind of goes on to that other question that somebody had asked. All right. One other thing I wanted to point out. Now, remember, again, the way it's set up, we talk about different types of signaling molecules, and then we talk about how they signal, right? What types of receptors do they bind to? And then what types of response is generated in the target cell, okay? So now we moved away from G protein coupled receptors to talk about another family of receptors called tyrosine kinase receptors, okay? And so these are very different, and some growth factors will bind to these types of receptors, okay? So that was another type of signal that we talked about, growth factors. All right, so again, trying not to get bogged down in the details, right? So now these pathways, like the JAK-STAT pathway, is actually part of cytokine signaling, okay? So cytokines was an immune molecule that I mentioned, and so they're going to bind to very different receptors. They're going to have very different responses to the cell, or initiate very different responses. So again, there's no way we can memorize any of this stuff, okay? Please do not memorize, just try to understand what's going on here. So this is, if you look at this pathway, this is a heck of a lot different than the pathway we looked at with the G protein coupled receptors. Do you guys agree? Okay, and a lot of it has to do with phosphorylation, and we know that phosphorylation in those other pathways as well, right? There's a lot of phosphorylation that goes on, because why? What does phosphorylation do? What does phosphorylation do to a protein?
Usually activates it.
Yeah, usually activates it. It can also deactivate it depending, but it alters the activity. So if you notice in all of these pathways, in different ways, things are phosphorylated, right? With those tyrosine kinase receptors, they can autophosphorylate each other, right? When it forms that dimer, and then that phosphorylation causes some sort of conformational change that activates the pathway. Here, this is showing you these particular proteins that become phosphorylated, right? Kind of directly from the receptor, and then they can form a dimer, and then where do they go? And what do they do in this case here? Where is this stat, right? This stat protein, when it forms this dimer, where's it going? What's this?
The
Nucleus. And doing what?
transcription.
Yeah, activating transcription, okay? So the themes are the same. It's just that the way it happens is very different. And again, you can see why these are really hard to study, right? Because there's so many different variations. And so if you wanted to target, you know, one specific molecule or something like that, the other thing is that likely that molecule will affect other pathways as well, right? So that's why these are hard sort of targets, but they're important targets, right? Because especially for cancer, Like I said, I mentioned a bunch of times of how these pathways are related, you know, to cancer, okay? This is again showing you another pathway, right? Where you see a growth factor binding to its receptor, and you got a whole bunch of other types of proteins here and a whole bunch of confusing stuff going on, right? What are these? Like MEK, MEK, and ERK? Those are kinases. What do kinases do? We just said it.
Immune cells.
Well, what do kinase, cytokines are proteins that are released by immune cells. Yes. But what do kinases do? Those are enzymes that do a specific thing. What do they do? What does it look like is going on here? What are these P's? Kinase is phosphorylate, right? Okay. So a lot of times in these pathways, you have different kinases that become activated. They then phosphorylate